I've got diverse fields to cover. Okay, thanks very much for having me. My name's Nick Jones. I'm a postdoc at University of Minnesota, and today I'm going to talk about some really cool research we've been doing, looking at what shapes phylogeography in puma populations across the rural to urban divide. Now, what really started our research was the essential question is we really don't understand how viruses move through a landscape through a host population. If we don't understand how viruses move, we can't understand how to control virus movement through a population. So this fundamental epidemiological characteristic transmission is pretty much unknown in wildlife, so there's a big gap. And this is particularly the case in urban areas. So urbanisation is the most persistent and long-lasting of all things we can do to an ecosystem, but we really don't understand how disease transmission is influenced by urbanisation. And this impacts species across the world, so it's a big issue. And probably part of the reason why we don't understand this is a lack of comparable populations with the same pathogen. And that's exactly what we've got here. We have a similar population in terms of number and demography of puma, but in totally different landscapes. One at the urban edge in Denver, so that's Denver you see there. You can see those mountains behind Denver, that's the Front Range. This puma population inhabits the Front Range and regularly forays into the urban matrix adjacent, so around Boulder as well, and up into Fort Collins. 400 kilometres away, we've got this other population of puma, which are in a totally different landscape, very much more natural, wildland, rural place, with a big mountain range in the middle called the Oncompodre Plateau. One day I'll get there, one day. This is a little bit more about our populations. You see up the top there, we've got Denver. Can I get that to work? Yeah, so that's in Denver. You can see the Denver area with the mountains in behind. Each white dot reflects one puma sampled. So there's a lot of white dots. And I just wanted to talk briefly about how much effort is involved to sample these puma. Like, it's enormous. So you can only do this in winter. You can only sample puma in winter in Colorado. You have to get a full dog team out, some vets, the researchers go out, they look for fresh trails. Once they find a fresh trail, puma trail, then they get a dog team onto that trail. The dogs chase the puma sometimes up to 75 kilometres. Eventually that puma will go up a tree. Then the researchers will catch up. They'll get a dart gun, dart the animal, and they'll set up like a big kind of, I suppose, mat underneath for the animal to fall into. Then they take that blood sample. And that's for one dot, so it's an enormous amount of effort. That's before the lab work even begins. So what we want to do with this data is address two really essential questions. So was the rate of viral spread similar in both populations? And are the landscape and host genetics shaping viral phylogeography in the same way in both populations? This is a bit about our data. So our host data was from tissue and blood samples collected from 2005 to 2015. We blasted it with double digest RAD sequencing to get about 20,000 SNPs per individual. And we had roughly similar numbers in both populations, UB being unbounded, WUI being the wildland to urban interface. We call it unbounded because in this population, these puma can really go anywhere. Where the WUI, they're kind of constrained by the urban matrix, if that makes sense. And you see just quickly looking at these populations from a host genetic point of view, they're really distinctive. Okay, there's a few potential immigrants in between these populations, but these two puma populations are quite genetically distinct. The landscape data we had was, we used something called a circuitscape approach, which basically treats landscapes as resistant surfaces. And these resistant surfaces are coded based on topography, roads, streams, etc. So basically, two individuals could be close in space, but if they're separated by a road, for example, they would have very high resistance values for roads. I can talk about this in more detail for anyone which is interested. And you can compare these resistant surfaces to just standard spatial distance or isolation by distance. So it's very cool. It's a very cool approach. And this is a particular virus we use, like a model virus called feline immunodeficiency virus. It's basically that human as a feline equivalent to HIV. It's really fast evolving, and it's generally, importantly, host specific. So it just transmits to puma to puma. Thought the transmission mode is meant to be, or is generally horizontal, so when they bite and scratch. But vertical transmission is possible between mother offspring and father offspring potentially as well. So we qPCR screened the same tissue blood samples we had for the host, and we basically got about 59 FIV genomes. So quite a big effort. Okay, so my position in this project was to link all these disparate data sets together, the host, the landscape, and the virus. And that's actually quite a complex thing to do, but one good thing you can do to start with is map onto the host phylogeny the virus phylogeny. This is what I did. These are a few of the details about my phylogeographic methods. I just will focus on the fact that in this population, we quickly worked out there's two subtypes co-circulating through this population, subtype alpha and subtype gamma. So two subtypes with very different evolutionary parameters. So that was quite interesting to start with. So very, very different viruses going around the same population. For the host, we use an SVD quartet approach to build a cladogram of the host, just based on the SNP data. And this is what you get. So a mess, a jumbled mess, a tangled mess, when you try to map the FIV phylogeny onto the host phylogeny in a co-phylogeny tanglegram type approach. You can see, I won't go into the detail of this, it's complex, but basically the host, the virus is on the right, the host is on the left. You see the colours at the end of the phylogeny, viral phylogeny. Those colours are reflected in the colours of the puma. So you'd expect if the host phylogeny mapped onto the viral phylogeny, you'd expect to see clusters of the same coloured puma silhouettes. And you clearly see that's not the case. There's a few examples of that. There's a few places where there's clusters of where the viral and host phylogenies match, but generally that's not the case. And what that means really is mostly these puma aren't transmitting to individuals which are related to each other, mostly. There is some evidence, and it's mostly in the woolly population in the urban edge, where we do detect some of that, but mostly that's not the case. Okay, so the other thing you have to do is actually put these phylogenies into the landscape and understand this in a spatially, temporally explicit manner. And now today, ladies and gentlemen, I'm going to show you a film, a quick film. It's going to be a box office sensation. Keep an eye out for it. It's titled Endemic Circulation Through an Unbounded Population. Take autographs later. And let me see if this works. I can't pause it. So just quickly, before we get into it, you see the lines there, hopefully? The lines on the landscape, the red lines in this case. That's a branch. And when the branches connect to the landscape, that's a node. And these nodes go to tips. And those tips are obviously red because they're infected. But we also show you uninfected and not tested samples as well. And each node is being positioned by the beast model we use to form all this. So it's spatially explicit and temporally explicit. Okay, are you ready? Okay, so you've got one subtype, and then gamma arrives, then you see it just jumping over the mountains. This big range in the middle, that big kind of black kind of dot, that's the big Oncompodre plateau splitting. So the virus is moving across, and importantly, not really forming a biogeographic pattern. They're really a mixed bag. There's lots of clades right next to each other. So it's not a clear geographic spread in this case. And we can talk about this for days. There's lots of interesting patterns here. Okay, now the exciting sequel to all this is endemic circulation through a population of the urban edge, and the pattern's really quite different, as you see. So again, this is just subtype gamma, subtype alpha wasn't found here. You can see it's spreading across the front range in Colorado, in the woolly population, across that urban edge. Even though the pumas regularly foray into the Denver matrix, or Boulder matrix, they're really not transmitting to each other in this case. And the FIV is moving up and down this interface here. So a very different pattern and much more spatial structure. If you look at it in detail, you see the clades kind of filter out geographically in this case. So quite a different pattern of spread in this woolly population compared to the unbounded population. Oops. Okay, and just to prove that these individuals do actually go into urban matrix, this is a classic video behind Boulder here. We've got a puma, probably trying to get a cat snap, I imagine. So definitely a big part of life.
Smell it.
Okay, you can watch the YouTube. It's amazing to see that. I've never seen one, obviously, but these guys just had one in their backyard. So it's pretty cool. Anyway, so these are just patterns so far, but what statistical tools was I going to use to actually quantify these? I use two techniques. I use a phylogeographic technique called Seraphim to address question one. This basically quantifies the velocity of spread to test how landscape and potentially host could shape the velocity of that spread. I also used a landscape genetic technique to address question two. It's called generalized dissimilarity models. Basically, GDMs are really cool. They're like a fancy, sophisticated Mantel test, which basically incorporates all the good things about a GAM. So it's a really powerful method. I highly recommend it. Basically, it's a way to link all these complex matrices together in a multivariate context. And basically, I used it to model relationships between landscape and host variables, host genetic distance, and to how it shapes viral phylogenetic distance. And what I mean by host genetics here is the proportion of shared alleles. What I found was spread at different speeds. So to address question one, we found that spread in both populations was quite different in terms of speed. So subtype gamma and subtype alpha in this population was spreading about 4.27 kilometres per year, where in the front range in the Wui population was at 4 kilometre less per year. This is statistically significant as well using a Bayesian approach. And compared to my previous work on bobcats using the same method in a very much more urban area, they were slower again, so about 1.6 kilometres per year for bobcats. So urbanisation, I think, is impacting the spread of these pathogens, which makes sense. And then if you look overall across all these landscape features and the phylogeography and spread, only high ridgelines are certain resistance factors to viruses. So across both populations, these big ridges are restricting the viral movement, which is quite cool as well. Okay, so part two, what shapes phylogeography? Are they the same or are they different? This GDM approach quickly shows that quite different factors shaping phylogeography in both populations. So you have the Wui population over there, the model structure, and the unbounded population. For the Wui population, spatial proximity and host gene flow was shaping the viral matrix. Okay, and the spatial proximity was actually nearly significant as well, but explaining quite a lot of deviance here. About 18% of deviance explained by these two factors alone. And that's compared to the unbounded population, where you get year, so there's a temporal effect, and there's also effect of roads as well, which is quite interesting. You think roads, that doesn't make sense. How are roads impacting gene flow of a virus or spread of a virus? And that's because roads in the unbounded population are small dirt tracks which the puma use to connect with each other. So if these two puma are connected by roads, these little paths, they're more likely to transmit theoretically. But that's not the case in the unbounded population, where spatial proximity was the most important, followed by host genetics. What does it all mean? So it means most transmission events occur between unrelated hosts, but perhaps relatedness is more important in the Wui population. Mountain ridges do actually restrict spread, which is quite interesting. But overall, in the Wui population, viral spread is down, isolation by distance is more important, but within an unbounded population, you get increased viral spread, but landscape host and spatial distance are less important. But hopefully what I've shown you today is basically these techniques can really gain really interesting high-resolution, multifaceted insights into how viruses get around landscapes. And with that, I've got lots of people to thank, lots of fantastic collaborators from Colorado State and other places, which this work wouldn't be possible without, and particularly these three people, Megan Kraft and Scott Carver. And with that, thank you very much. Any questions before I get going?
The dispersion of the virus seems to be dominated by the males, right? That are like, is that males the ones that got the long lines that
potentially
go the range?
Like sex wasn't important in the models, which is interesting. So we're expecting possibly a sex effect, but that didn't come out at all, which is interesting. But generally, yes, males are the dispersers. They disperse at long distances, but we didn't detect that necessarily so far. But we're still digging into this data a bit more, so maybe a sex effect will come out. So, good question. No? Thank you very much.